Hi everybody, it's Sahar again, and today I'll be doing chapter 32. I'm only going to be doing half, Rachel will be doing the other half. So this is just 32.1. So this is just 32.1 and 32.2. So 32.1 is what is a protozoan, and 32.2 will be talking about what are the major groups of Lophotrochozoans. So as I said, 32.1 is what is a protozoan. Now if you remember from the last chapter, there are two types of diploblastic clades, and that's protozome and deuterosomes. And protozomes have far more species than individuals. And in most protozomes, the embryonic blastospore becomes the mouth. And again, going back to the last chapter, a triploblast is something that has a mesoderm, an endoderm, and an ectoderm. So this is very much linked to the last chapter, so it would be a good idea to go and review back if you don't remember everything. Okay, so protozoans are bilaterally symmetrical. They have an anterior brain that surrounds the digestive tract. So again, like in the last chapter, we had seen the anterior brain, and it had the picture of the fish. So all the sensorial organs were in the front of the organism. They have a ventral nervous system with paired or fused longitudinal nerve cords. So the protozoan clades have modified coeloms, and arthropods, for example, they don't have a coelom, and their body cavity is now called a hemocoel. On the other hand, we have mollusks, who have an open circulatory system, but retain vestiges of an enclosed coelom. So this is part of the blood chamber, which is a part of the open circulatory system. Down here, if you look, here's a picture of, this is also in the last chapter, of where the coelom would be, and this is again the triploblastic, so it's the ectoderm over here, and then you have your endoderm inside, and that would be your mesoderm in the middle. So here's just a table for anatomical characteristics of the major groups of protozoans. So you don't really need to know this, but you can just look at it for your own reference. And as it says at the bottom, all protozoans have bilateral symmetry. So protozoans were split into two clades long ago, and they have been evolving ever since. So this is divergent evolution. And the two clades are Lophotrochozoans and Ascidiozoans. Sorry for my pronunciation. And here's a picture of what it would be like. So we're going to be going into some of these things. And yeah, just kind of know it roughly. Here are some characteristics of Lophotrochozoans. So they have internal skeletons, and many of them have free-living trochophore larvae. And several groups have a lophophore, which is a circular or U-shaped ridge around the mouth with two rows of hollow ciliated tentacles. So you can see here, these would be your hollow ciliated tentacles. So the lophophore is thought to have evolved twice, or it can be considered as an ancestor, and it has been lost by some groups. Most animals with a lophophore are sessile as adults. So this would be an example of ectoproct, brachiopods, and phoronids. And again, sessile means that they do not move, so that means they're like ground in. They don't really have limbs or anything to move around. So yeah. Some Lophotrochozoans have spiral cleavage, and again, this is something that we went over in the last chapter. An example of this would be flatworms, ribbon worms, annelids, and mollusks. And we'll be going into depth with all of these later. And many of these are worm-like structure. They have worm-like structures because it is advantageous for them to burrow into soft sediments. So if you know about like mollusks or flatworms, even worms, they all kind of go into the soil. That's how they protect themselves, and some of them, that's even how they obtain their food. So mollusks are the most species-rich group and have a very different body plan. So here's a quick fun fact about that. Cuttlefish are not fish, they're actually a type of mollusk. Even though they have what they would consider kind of like a shell around it, it's actually inside the body, and it's used for buoyancy more than it is for the actual reasons a shell is used for in a mollusk, for example, which we'll be getting into later. Ascidozoans have an exoskeleton, and this is a non-living covering secreted by the epidermis. For the animal to grow, the exoskeleton must be shed or molted and replaced with a larger one. We had briefly talked about this in class a couple times. Miss J mentioned it. It's basically when, for example, when a crab grows inside its soft tissue, which is inside, it's going to expand. But the exoskeleton is a hard shell, so that can't really grow. So what the crab has to do is that it has to basically come out of its shell and regrow a new shell. So molting has evolved 500 million years ago, and apparently it's only evolved once. So you don't really need to know that very much, but just kind of like, we'll be talking about like a lot of when they evolved and when what evolved. You don't really need to know it, but it's just better for you to graph out basically the evolutionary pattern of all these organisms, which is just for a better understanding, basically. So here are some pictures of molting. So on this side, you can see this is a fossil, and you can see this was the emerging animal, and this was the molted exoskeleton. So this is the animal kind of like coming out, and this would be its hard shell that's leaving behind. It's actually kind of cool that it got captured like this, because this is like half in motion, and then it just kind of died, I guess, like that. And here's a picture of a spider, I believe, that has molted. It's actually pretty creepy, but this is what a spider looks without its exoskeleton. And this would be its exoskeleton. So before molting, the new exoskeleton is already forming, but it's not hardened yet. So after the old one is shed, the new exoskeleton expands, and then it hardens. But before it hardens, the animal is really vulnerable to predators, and the exoskeleton is soft, and the animal cannot move much. So this is going back to the fact that muscles are attached to the exoskeleton. So to move, the exoskeleton plays a big role in that. The organism can still move without the exoskeleton, but it does not have a full range of motion. So for example, it would almost be like if we didn't have bones. Of course, if we didn't have bones, we would basically be able to do nothing. But these organisms, they can do a few limited motions, but they can't be as fast or be as agile as they would with an exoskeleton. So that makes them vulnerable. Some ascidozoans are worm-like and covered with a thin exoskeleton called a cuticle. Now this is not to be confused with plant cuticles. Plant cuticles are, as we have covered, something that is a waxy layer on top of leaves, but this is a cuticle that's on an organism. So make sure that you don't mess these up. Cuticles allow gas and water exchange, but they restrict animal to moist habitats. So for animals to be able to move, and if they have a cuticle, they have to be in moist habitats. So like if you look at worms, which have a cuticle, they have to be kind of in moist soil, or that's why they go nice and deep, where they can get some nice moist soil. Other ascidozoans have a hard exoskeleton, and this is thickened by proteins and chitin. And as we had learned before, chitin is a strong polysaccharide that comes from glucose, and it's also seen in fungus. So these animals have appendages for movement, and that would be classifying them into the arthropod clade. And the appendages are used for a couple different things, and for example, it would be locomotion, food capturing, gas exchange, copulation, and sensory perception. Muscles are attached to the inside of this exoskeleton. So I'd said this before with the crab and the spider that we saw when it was molting. The hard exoskeleton allows the organism to walk on dry land, and it prevents the organism from drying out. This is a major piece in evolution. So this is what could have led to aquatic arthropods coming out of water and starting to become land creatures. And as I said before, worms have cuticles, which is an exoskeleton, but they dry out because of it. That's why they need to be in moist areas. But if you organisms that have hard skeletons, they don't dry out. So that's why they can leave the water and come onto land. And evolution, yay. So here's a picture of what it would look like inside of an exoskeleton. So you can see all these muscles attaching to your exoskeleton. So these would contract and relax to pull these appendages up and down, right and left, however needed. Now we're going to go into a class of organisms called arrowworms. And these have evolutionary relationships that have been long debated, so nobody really knows exactly how they've evolved. So early development is similar to the deuterostomes, but these are features ancestral to all triploblastic animals. So we can't really classify by that because many, many species have that. Their gene sequences that identified them as protostomes, and they may be a sister group to all other protostomes. So we're not sure by 100% that they are protostomes, but they could be a sister group or they could be protostomes themselves. So here's what a arrowworm looks like. You have like the head, and they're kind of separated into three parts: the head, the trunk, and the tail. Yeah. So arrowworm columns are divided into three compartments, and as we'd seen in the previous picture, it was the head, the trunk, and the tail. So the body is transparent, and most of them swim in the open sea. Now, because they're transparent, they can swim in the open sea. Otherwise, if they had any pigmentation or coloration, they would become pretty easy prey in the open ocean. So here's another thing in regard to evolution and being able to map out what animal diverged when. We can see fossils of arrowworms from 500 million years ago, so they've been along for a really long time. And this has kind of been taken into account for trying to figure out where these arrowworms have been evolved from, but as we talked about, we don't really know where they came from. So arrowworms are small enough that gas exchange and waste secretion occurs by diffusion through the body wall. So they don't really need any gills or lungs or any other type of specialized organ, because they can literally just do all the functions that a cell that our different systems do through a cell. So they don't need an excretory system or a respiratory system. Their cells can just do it by themselves. They are stabilized in water by two pairs of lateral fins and a tail fin. So this was seen in the picture right here. So you can see the lateral fins are right here. They have an anterior set and a posterior set, and then they have their tail fin. And this is what helps them balance and basically what helps them move. And their prey is planktonic organisms. So here's a question on arrowworms. Which of the following statements regarding the arrowworms is false? Now, I know it says shown, it's not supposed to, it's not shown here. So, A, they cannot be clearly classified as lophotrochozoans or ascidozoans. They have a body plan based on a coelom divided into a head, trunk, and tail. They have a complete digestive tract. They lack a circulatory system and have cilia that line the coelomic cavity. Take a second to try to think what it is. If you want, go back in the podcast a bit. And if you can't figure it out, at least try to do process of elimination. If you thought that the answer was C, then you're right. So it's gone. Here's another question. Which of the following statements regarding cuticles in ascidozoans, such as that found in the Pripolida, is false? A. Cuticles protect the animal. B. Cuticles provide support for the animal. C. Cuticles, in conjunction with longitudinal muscles, provide a mechanism for movement in the animal. D. Cuticles allow the exchange of gases. Or E. Cuticles allow the exchange of water. So again, take a second to think about it. And the answer is B. If you look at the other choices, most of them describe an overall exoskeleton, like a hard exoskeleton that we see in crabs. So in conjunction with longitudinal muscles that provide movement. So that is talking about how muscles are attached to a hard exoskeleton for movement, and that's not seen with cuticles. Cuticles is a soft exoskeleton. Alright, so now moving on to 32.2. And this chapter is about what are the major groups of Lophotrochozoans. So here is a diagram, and we'll be going into a lot of these on this side. So they come from ectoprocts. So ectoprocts are bryozoans, or moss animals. So ectoprocts are colonial, and the external body wall secretes material that form a house. So they're basically like their extracellular matrix, and they excrete certain things that allow them to kind of clump together and become one house. Now they're really small. Their individuals are probably from 1 to 2 millimeters in size, so that's really small. And they're all connected by strands of tissue, so that's how they kind of stay together. And a colony is secreted by asexual reproduction by the founding member. So there's no sexual reproduction that takes place when making a colony. Colonies may contain as many as 2 million individuals. So here's a picture of it, and this would be a colony actually. This is not just one of them. So you can kind of see all the little ectotrophs all clumped together. So individual ectoprocts can rotate the lophophore to increase contact with prey. Now remember what lophophore is? They're the ciliated, and they're kind of like little arms that flail out. And they can also retract them in. So they can either take them out to grab prey, or they can bring them back in. So ectotrophs can also reproduce sexually, but the colonies themselves are produced asexually. This is done by sperm that is released into the water and carried to the other individual. And by individual, it would basically be probably some other ectoproct in another colony. And then the eggs are fertilized and brooded internally, and then the larvae are released. So individual ectotrophs can Alright, so now moving on to flatworms, rotifers, and ribbonworms. So flatworms, rotifers, and ribbonworms form a monophyletic group, even though they have different body plans. They have other things that are similar between them that classify them in the monophyletic group, but their body plans are actually pretty different. So we'll get into those. Flatworms do not have a system for gas transport. Every cell must be near the surface, and this is accomplished by dorsoventrally flattened body. And that's why they're hence called flatworms, because all the cells are near the surface so that gas exchange can take place. Their digestive tract is a mouth opening into a blind sac. The sac is often branched, and it increases surface area for absorption of nutrients. So we'll take a look at a picture of it. So here you can see on the side it's a flatworm. This one's really pretty. It has like different colors and everything. And here you can see the gut. So the gut is this dark brown in the middle, and it branches off on the right and the left, and that's what it meant by branching. And this is what allows for the high surface area for more absorption. And here would be the opening on the bottom. So small free-living flatworms have cephalization, and this allows them to glide over surfaces by broad bands of cilia. So if you saw in the last picture, it kind of looked like it was floating around, and that's what allows it to do that. Turbellarians include most free-living species, so that's what they can be classified under. Most flatworms are internal parasites, and tapeworms and flukes, so those are examples of them. The one that we saw before, that was, I believe, an aquatic flatworm, but a lot of the other flatworms are what they live in digestive tracts of other organisms. And they absorb digested food from the digestive tract of the host. So here's another question. Which of the following statements about flatworms is false? A. Flatworms that can move do so by ciliary action. Sorry for my pronunciations again. B. Flatworms have a complete tubular digestive system. C. Flatworms depend on diffusion to accomplish the functions of a specific circulatory or respiratory system. D. Most of the species of living flatworms are parasitic. E. Parasitic flatworms, such as tapeworms, frequently lack a digestive system. So take a second to look at it and decide what you think it is. Alright, so the answer is B. Flatworms have a complete tubular digestive system. And that's false because, as we had gone over, their digestive system is branched, and that's what allows for the high absorption and the high surface area. Now, some of these flatworms can cause serious diseases such as schistosomiasis. And the monogeneans are external parasites of fish and other aquatic animals. So that's a type of tapeworm. Now, moving on to rotifers. Now, these are also really small. They're 50 to 500 millimeters, but they have specialized internal organs. And they also have a complete gut. Their body cavity is a pseudocoel, and it functions as a hydrostatic skeleton. But most propel themselves by cilia. Even though they have a hydrostatic skeleton like we see in worms, they usually move by cilia. The corona is a ciliated organ on the head, so that's one of their specialized organs, and it basically has beating cilia that sweep food particles into the mouth and into the mastax. We'll look at that in the diagram. And the mastax is an organ that grinds the food, so it's, I guess you could say it's equivalent to our teeth, and the cilia sweeping, I guess, would be like our arms. So we're bringing the food into our mouth, and then our teeth are grinding the food. Some can protrude the mastax through the mouth and capture prey with it. So they don't have to use their cilia, they can also just use their mastax. Betyloid rotifers have only females, and their eggs develop without fertilization. That's actually pretty cool. They don't have to fertilize, they just kind of develop. So here's a picture. So this is where that mouth is, and this would be your mastax over here. That's like your grinding system. And here are your cilia that would kind of sweep everything in. And they're actually fairly long. If you can see in this picture, this is the main digestive tract down the middle. And you can faintly see the rest of the body. And these are the cilia that are protruding out from the body. So here's another question. Which of the following structure is not present in the rotifers? A. Corona. B. Mastax. C. Pseudocoel. D. Segment. E. Complete tubular digestive system. Take a second to think about it. It's D. Segments. So again, as we'd gone through, the corona is the cilia on the head, the mastax is the grinding system, they have a pseudocoel, and they have a tubular digestive system. Now we're going to get into ribbon worms. And they're also I'm going to butcher this, nemerteans. So here are their characteristics. They have a complete digestive tract too. They have a fluid-filled cavity called a rhinocoel, and inside is a hollow muscular proboscis. Now, if you've noticed a pattern yet, anything that has coel refers to a body cavity. So a pseudocoel, a rhinocoel, and we'd also seen some other examples in the previous slides. So muscle contractions cause the proboscis to be expelled explosively from an anterior pore. And they may be armed with sharp stylets to pierce prey and release toxins. So this is one of their defensive mechanisms, and we'll look at that in a diagram. So this is what a ribbon worm looks like. And you can see that this is the proboscis right here, and this is the retracting muscle. And as soon as there's some danger or any other event, it'll protrude out and it'll come out, and that can hurt. It can have toxins. It can be used to sting or basically knock out prey. And they actually have a pretty simple body plan, because it's just one tube here for the proboscis and then just one tube for the digestive tract. So now we're going to have a quick break time because we're halfway through the PowerPoint. So I'm going to show a quick video just to relax everyone, and yeah, enjoy.
So you've heard many of life's curiosities, but did you know that cashews grow on apples? In this case, the apple is simply an accessory fruit with the cashew growing on top. Or how about the fact that mushrooms are more closely related to animals than plants? The most common ancestor between humans and mushrooms is more recent than that of mushrooms and plants. Trees, they actually grow out of the air, not the ground. Though they receive some minerals from the soil, the carbon dioxide from air goes into the tree and with the help of the sun removes the oxygen. This carbon is left with water to create the substance of the tree. And did you know that there's enough water in Lake Superior to cover all of North and South America with 1 foot of water? Or that there are more stars in the visible universe than all the grains of sand on all the beaches of Earth? And yet the number of synapses in your brain's cerebral cortex alone, about 125 trillion, is the amount of stars that would fill 1,500 Milky Way galaxies. But an atom is around 99.99% empty space, making everything in the universe mostly nothing, including your body, which, by the way, is constantly replacing its cells. So much so that every 10 years, your entire body has replaced itself. Every atom in your body is different now, meaning you have no more in common with your previous self than any other human being that's lived or will ever live. All the while, there are more bacterial cells in your body than actual somatic body cells. And if you've made it to age 18, you have, on average, 3,200 to 3,500 weekends left in your lifetime. More importantly, you only have about 57 summers left. And did you know that while dragonflies have legs, they can't walk, only land? Or that in a population of clownfish, if the female dies or is removed, the most dominant male will become a female? The Tyrannosaurus, in terms of time, is closer to today than to when the Stegosaurus roamed the Earth. To the same token, Cleopatra lived closer to the first moon landing than to the construction of the pyramids. And just for fun, when I say it, you are now breathing manually. Got a burning question you want answered? Ask it in the comments or on Facebook and Twitter, and subscribe for more weekly science videos.
Alright, so now before we go back to work, feel free to comment below on things that you agreed with and things that you can counter, or what things that you actually knew and things that you found surprising, and any other facts that you would think were interesting and you would like to share. Alright, so now we're going to continue on with our second monophyletic group, which includes brachiopods and phoronids. And they also have lophophores. And they may have evolved independently from the ectoprocts, but we don't really know exactly. Phoronids are small, sessile marine worms. So again, sessile means that they cannot move. And they secrete a tube of chitin in which they live. So they basically kind of make their own little house and they stay in there. In most species, their eggs are released into the water and they're fertilized. But few species are fertilized and brooded internally. So you can have both ways. So here's a picture of them. And this would be basically your outer covering of chitin, which would hold it into the ground. And then these would be your lophophore tentacles. And again, they're also pretty simple. It just they have one digestive tract like this, and it just goes out. So this is the mouth, and that's the anus. So here's another question. Which of the following are true about phoronids? A, they are species rich. B, they secrete tubes made of chitin. C, they are free living. D, they do not have a lophophore. E, they are greater than 50 centimeters in length. So take a second to think about it, or you can go back, you can pause the video. Alright, so the answer is B. They secrete tubes made of chitin. They are not very species rich, they don't have that many species, and as we know they're sessile, so they cannot be free living. And they do have lophophores, so that's true. And they are not greater than 50 centimeters in length. They're actually smaller. Most of the organisms that are lophotrochozoans are pretty small. A lot of them, as we've seen, are smaller than even 1 centimeter. So now we're going to talk about brachiopods, which are solitary marine mammals. So they're not exactly sessile. They can kind of move around, but they don't really move to and fro. They kind of just move their body parts more. So they have two-part shells, which are connected by ligaments. They resemble bivalve mollusks, but their shells have evolved independently, so they're not exactly mollusks. They have two half shells, which are a dorsal and a ventral shell. So we'll look at that in a picture. So this would be your brachiopod, and you can see your lophophores inside, and these are your two shells. And usually dorsal refers to the top, so this is the dorsal, and that's your ventral. So brachiopods live attached to a substrate, and some species have a stalk. So, as I said, they don't really move to and fro. They are not exactly sessile because they attach themselves to something else, but they can move their body parts more. They do produce sexually, so their gametes are released into the water for fertilization, just like all the other marine animals that we've talked about that produce sexually. And their larvae are planktonic, so they're really, really small. Brachiopods reached peak abundance in the Paleozoic and Mesozoic. So again, that's one of the facts that just kind of help you map out the evolution of organisms, and it could also help you separate the organisms in your mind. There are only 355 species that have survived. So there were quite a few back then, but we don't exactly know why, but now we only have a couple that are alive today. So now we're going to go on to our last monophyletic group, which are annelids and mollusks. So annelids and mollusks are sister groups. So annelids have a segmented body, and their column in each segment is isolated from the column in the other segment. And they have a separate nerve ganglion that controls each segment. So basically, if you think about one of those buses that are actually like a couple different buses put together, each part is segmented. Each part has a different door. Each part has a different seating. Each part has everything differently, except they're one long bus. So most have a thin, permeable body wall that serves for gas exchange, but it's restricted to aquatic or moist habitats. So here's a picture of an annelid, and you can see this is a ganglion in the ventral nerve cord, and these are your segments, these little parts, and each one is served by a different ganglion, and you can see, as I said, you can see all the segments. And if you look here, you can see when you cross-section it, where the nerve cord is, where the blood vessels are, and the longitudinal muscle, and the column, of course, too. Alright, so here's a question. Which of the following is false about an annelid? A. They have a thin, permeable body wall. B. They have segmentation. C. They need to live in moist environments. D. Each segment has one ganglion. E. They have one ganglion for the whole organism. So take a second to think about it. Alright, so the answer is E. They actually have one ganglion for each segment, basically. So now we're going to go on to Polychaetes, which are mostly marine animals, and they burrow themselves in soft sediment. They have one or more pairs of tentacles for filter feeding, and that was, again, something that was covered in the last chapter. Parapodia on each segment are extensions of the body wall, and they function in gas exchange and sometimes for movement. So we'll see these things in the picture. So here are your tentacles that come out for filter feeding, and they have these little, basically you can say hair-like structures, and that's what allows them to filter their water for food. And these would be the extensions from the body. So pogonophorans are polychaetes that have lost their digestive tract, and they secrete tubes of chitin, which they live into. So basically, they are similar to the organism that we've talked before, but they don't have a digestive tract. How they digest or take in nutrients is by dissolved organic matter from their substrate. And this is done by facilitated by endosymbiotic bacteria in a specialized organ called a trophosome. So they don't really get their nutrients from themselves, but this is also not considered parasitic. So here's a picture of them. I think these were in Finding Nemo. You saw these little red things at the bottom flailing around. Yeah. So Pelagoniforms were discovered in the 20th century in the deep oceans, and they were found living in high densities. The largest one that we've seen of them are up to 2 meters long, and they live near hydrothermal vents. So they can get really long. And their endosymbiotic bacteria fix carbon using energy from oxidation of H2S. So this is where they get their nutrients from, and that's the endosymbiotic relationship with their bacteria. Celtolates are freshwater or terrestrial. So that's another organism, a class of organism that we're going into. And they're all, I'm going to butcher this again, oligochaetae, and they're also leeches. So oligochaetae have no parapodia, eyes, or tentacles, and they have few setae. Earthworms, for example, they bury in and ingest soil, so they are an example of oligochaetes. All oligochaetes are hermaphroditic, so that means they're both male and female. And we talked about this during the reproductive chapter, in which worms have both the female and the male parts, and how they copulate with another worm. So each individual gives and receives sperms during copulation. So this is the worms that we were talking about and how they are hermaphroditic. So leeches also lack parapodia and tentacles, and leeches are freshwater and terrestrial. Their column is not divided into compartments as we would have seen in the oligochaetae, and their segments at either end are modified to form suckers. So they have it on both ends. And they also serve as temporary anchors that aid in movement. Here's a picture of a leech, and you can see that there's a sucker on one end and on the other end. And this is a fairly pretty one, though. We know the stereotypical black leeches from the movies and the Hollywood scenes, and yeah, so they can also be colorful like this. Leeches make an incision in the host and secrete an anticoagulant into the wound. So this is how they basically feed, and these are considered parasitic. But we do use leeches in medicine, and they're used to treat diseases that are believed to have caused excess blood or bad blood, and this was seen in the medieval times. A lot of the therapies for any disease, or for sometimes the bubonic plague and such, was to use leeches or to drain out blood, because it was considered that the disease was caused by bad blood. And now we know that that's not the true case. So Hirudo medicinalis is still used today to treat wounds and prevent scarring, and that's a type of leech. So now we're going to go to mollusks, which have a body plan that has 3 main components that we're going to talk about. So they have a foot, which is large muscular structure originally for locomotion and support of internal organs, and the foot has been modified into different groups. So this is basically their strength of the whole mollusk. They have a visceral mass, and this includes the heart, the digestive, the excretory, and the reproductive organs. So these are only 2. We're going to be going into the third one, mantle, later. But basically, the foot is the muscle part of it. The visceral is the soft part of it, I guess you could say, like the part that doesn't really aid in movement. So mantle is the third part, and it's the fold of tissue that covers organs in the visceral mass. And the mantle secretes the calcareous shell, so that's what leads to the formation of a cell. And in most species, the mantle extends beyond visceral mass to form a mantle cavity. And this contains the gills for gas exchange. So here's a picture of it, and this is a generalized molluscan body plan. So at the bottom is, so at the bottom we see the foot, and this is the heart, the digestive glands, and this would be your digestive tract over here. And your mantle is highlighted in orange. It's not the actual shell. The mantle is not the actual shell. The mantle secretes the shell, but it's not the actual shell. And yeah, so this would be your visceral area, and this is your mantle on top and your foot on the bottom. So it's kind of like a sandwich. And here's the more complex formations, and this is an echidna. So you can see that the gills in the mantle cavity down here, and this is the foot, and this is the digestive tract. Again, this is still the visceral mass with the digestive tract in the middle and the digestive gland in the middle. And then your mantle kind of wraps around this time. It's not just sitting on top, it's like wrapping around. And these are your shell plates. Now this organism has shell plates instead of one solid shell. And this is a gastropod. And again, it's a different type of mollusk. So you can see the foot at the bottom. And now you can kind of see where the idea of a foot came. It kind of looks like a foot right here. Oops. And this would be their visceral mass over here. And you have your mantle right here, this orange part. And again, it's wrapping around, not just sitting on top like another one. And this whole thing is your shell. And here's another example, another two examples of mollusks. So this is a bivalve. So this is what we were talking about when we referred to the bivalve mollusks. So here is your digestive tract right here. And here are your gills. And then this would be your mantle. It goes all the way from the bottom and it goes all the way around and then it comes to the top. Then the shell's on top. And as you can notice, it doesn't really have a foot. And then we have cephalopods, which also don't really exactly have foot, but this, I guess, would be considered your foot because that's what aids in the movement. Here's your digestive tract and your heart, so that's your visceral mass. And then your mantle sits on top right here, and then your shell is there too. So yeah. Now cilia on gills beat to create water current, and this is what allows for them to have a more efficient way of gas exchange. So the gills have many blood vessels for gas exchange, and we'll be seeing all this in the respiratory system when we learn about the respiratory system. So some species use gills for filter feeding, and others have a rasping structure called the raduli for scraping algae from rocks. So either they use filter feeding or they use, like, I guess you could consider it mechanical feeding, where they actually scrape the algae off rocks and they eat them. Mollusks do not have a closed circulatory system though. Their blood and fluids empty into the hemicoel, where oxygen is delivered to the internal organs, and blood eventually re-enters the blood vessels and is pumped by the heart. So basically, as Ms. J had said in class, it's like marinating your tissues. You just have the blood kind of floating over it. There's no exact vessels or exactly like an artery and everything like that. It's just there. And then it all circles back to the heart, and the heart just pumps it back, and it just kind of sits, and it comes back. So here's another question. In the mollusks shown, which of the following structure partly encloses an external cavity in which the gills lie? So, is it the A, radula, B, foot, C, mantle, D, coelom, or the E, visceral mass? So you can go back to the diagram. There's no diagram in this slide, so you can go back to the diagrams, you can look at the pictures, and come back and try to answer this question. So I'll give you a couple seconds. Alright. So the answer, wow, I really can't speak today. The answer is the mantle. Now we're almost done, so we're going to be finishing off with a couple more groups of organisms. So we're going to now go on to the monoplacophorans. These are the most abundant mollusks during the Cambrian, and only a few species survive today. So again, this is one of those evolutionary facts that are found in the book, and it's more just for better understanding. You won't really be, there's not many questions that are going to ask which came first, which came last, but there could be one or two maybe on the test. So they resemble limpets, and those are organs that are repeated over the length of the body. So they're kind of like, they look like a repeated organism, basically. So chittons are another organism that have multiple gills and shell plates. And most are marine herbivores that scrape algae with a radula. So that's what we had talked about before. And here's an example of an organism that has a radula. They cling tightly to rock surface with a large muscular foot. So here's a picture of one. It's actually really pretty. And this is the top of it. So at the bottom is how it would be like holding on with its foot. And maybe this light red color that you see around is probably the foot oozing per se, oozing out. And these would be your shell plates on top. So now we are going to go into bivalves, and those include clams, oysters, scallops, and mussels. They have hinged two-part shells, and many use the foot to burrow into mud or sand. And they feed by bringing water in the current siphon. So basically they rely on the movement of water for food. And filter food particles with large gills is basically what the in-current siphon is. And the water exits through the excurrent siphon. So it's like a gut. It kind of comes in through one end, the food gets filtered out, and the rest of the water that doesn't have anything pertaining to the food exits through the excurrent siphon. So here's the picture, and it's a large, I believe it's a clam. It's really big, and you can see this would be the opening right here. And they can grow fairly big, and you can see compared to the scuba diver in the back, it's huge. So now we're going to go on to gastropods, which are snails, slugs, nudibranchs, which are basically sea slugs, limpets, and abalones. And most of these move by gliding on the foot, but they can also serve as a swimming organ in some of these organisms. So not only can they be just for, you could consider, I guess, walking, but they don't really walk, but they can also be used for swimming. So nudibranchs are organisms that have lost their shells. And many are toxic and have brilliant colors for warning. So that's called aposematic coloration. So here's a picture of a sea slug, and it looks pretty crazy. It almost looks like an alien. You can see the different coloration in it and the bright colors that serve as for the aposematic coloration to warn that they are pretty venomous. You can see the blue coloring down here. And here's a quick picture of something called a blue slug, and they look really pretty. As you can see, they look really pretty, but this coloration is to show how venomous they are. And their tentacles actually have a lot of venom in, and they can actually kill a person if you get stung by them or you hold them for too long. But of course, there's always somebody who's still going to hold them. So. Alright. So now we're going to go on to land snails and slugs, which are the only mollusks that live in terrestrial habitats. The rest live underwater. The mantle tissue is modified into a highly vascularized lung. So instead of having gills that would be near the foot, they now have a mantle tissue that has vascularized lungs. So here's a picture of your average snail, I guess. And this is where your mantle would, this is where your shell is, so your mantle would be kind of down here. Now we're going to go into cephalopods, so octopus. So those include squids, octopuses, and nautiluses. So they also have an excurrent siphon, but it's modified. And the animal can forcibly eject water from the body cavity, which allows them to move by jet propulsion. So it's kind of like the nitrous at the back of a car. It's what lets them kind of shoot forward. That's what gives them that like kind of bobbing motion where they kind of go forward and then relax and then go forward. So they've become dominant marine predators in the Devonian, but now they are kind of, I guess you could say, in the middle of the food chain. They're not really on the top. So here's a picture of a cephalopod. This is an octopus, of course, and you can see all the tentacles. 1, 2, 3, 4, 5, 6, 7, 8. Yeah. Some cephalopods have a head with a complex sensory organ, so that's why octopi have this huge head, because that's where all their sensory organ is stored, because these are basically just limbs. They don't really have much except for nervous and circulatory tissue. And that's why they also have really big eyes. If you've ever seen a giant squid eye, I believe the size is as big as a softball. They're huge. Or even bigger, it might have been. So their eyes are comparable to those of vertebrates. That's for a normal sized cephalopod. But, of course, the giant squid is an exception. The head is associated with a large branched foot that bears the tentacles. So those are your 8 feet that stick out. And many early cephalopods have chambered shells. So they don't have shells anymore, but they used to. And they used to be chambered for different parts of the body. And those where the Nautilus is the only one with an external chambered shell that survives today. And it's these. I bet everybody's seen these sometime or the other in their lifetime. But that's what they are. They're actually Nautilus, and this is an outside shell which is chambered. So yeah. So that's the end of 32.2, and that's the end for my part for chapter 32. And here are my credits. So it's the biotextbook, the PowerPoint from Ms. J, the biology website, the images, and ASAP Science. Now I'd highly suggest everybody to go back to the bio website and just go through the summary that they have and just watch any of the tutorials, because those will really help you understand everything, because I can't show everything on the podcast. So I would really suggest everyone to go ahead and look at all the tutorials in the images on the website. So that's the end. And again, as always, I'm going to leave with a relaxing video. And again, it's another one from ASAP Science. So enjoy.
We all look forward to the future, but have you ever realized that we only see the past? It actually takes 80 milliseconds for your brain to process information, meaning we're never truly living in the now. Not to mention, it takes time for light from an object to reach your eyes, so technically, you see how they looked a fraction of a second ago. Similarly, when you look up at the stars, you're actually seeing light that has taken years to travel to Earth, thus seeing how the stars looked in the past. The next time you're looking, you may want to consider that that light has traveled all the way through the universe until it bumped into you. So watch out. Or not, because seconds ago you were miles away. How? Well, the Earth is rotating while simultaneously revolving around the Sun. But the Sun is also spinning around the galaxy, which in turn is flying through the universe, making your chance interaction with that light pretty incredible. It sort of puts the size of things into perspective, like the fact that a dust particle is roughly halfway between the size of Earth and a subatomic particle. Meanwhile, there are more atoms in a glass of water than there are glasses of water in all of Earth's oceans. Speaking of oceans, if all the oxygen on Earth disappeared for just 5 seconds, the ocean's hydrogen atoms would be free and evaporate completely into space. By the way, you can't actually see the Great Wall of China from space. You can, however, see the air pollution. Feeling disoriented? If you travel north, you'll eventually go south, but if you travel west, you'll always be traveling west. Travel to buy a lottery ticket, and your chance of dying on the way is actually higher than winning. Meanwhile, when a pregnant woman suffers organ damage, such as a heart attack, the fetus sends stem cells to the damaged organ to help repair it. That made you smile, because even faking a smile stimulates the same part of the brain as a real one, which can trigger endorphins and happiness. At the end of the day, humans are really just machines that turn energy into ideas. And it turns out that all of history and time has led to you listening to these ideas right now. So thanks for your time. If you want even more facts, make sure you're following our Facebook and Twitter, where we post new science stuff every day. And if you want the inside scoop on upcoming episode ideas and behind the scenes, check out our personal Instagram and Twitter handles. And subscribe for more weekly science videos.
Alright guys, hope you guys enjoyed all the podcasts. And again, put in the comments what you liked about the videos, what you liked about the podcast. and tell me something that maybe a fact that you know that not many other people know. So have a good day and bye.